My name is Meena Fatah. I'm a graduate student under the supervision of Dr. Robert Hamilton and Dr. Mike Seed at the Hospital for Sick Children, and under the supervision of Dr. Hamilton again and Dr. Yusen under the Answer Create HeRo program. So today I'll be talking about my project, which is elucidating the role of autoantibodies in the pathophysiology of Brugada syndrome. Next slide, please. Brugada syndrome is an inherited rhythm condition. It has a prevalence of about 1 in 2,000 and is responsible for about 28% of sudden cardiac deaths. The current gold standard for diagnosis is the Shanghai criteria, which diagnoses individuals across a series of clinical categories, of which in the center of the screen, the ECG that you see is required for a diagnosis. Unfortunately, though, this ECG manifestation is transient and not always present at rest or baseline. And in some individuals, they have to be given drugs to bring this ECG manifestation out. Adding to the complexity of the disease is its genetics. It's inherited in an autosomal dominant and incompletely penetrant manner, meaning that where an individual has a pathogenic mutation for it, they have a 50% chance of passing it on to an offspring. But even when an individual has a mutation, it doesn't necessarily mean that they'll develop disease. And the only known pathogenic mutation only accounts for about 30% of cases. So in the other 70% of cases, we wondered, could it be novel genetics or secondary pathologies that are at play? Next slide, please. So in the far left, we've actually published in June of 2020 that indeed there are secondary pathologies at play. We discovered autoimmunity in individuals with Brugada syndrome, which is essentially their immune system creating antibodies against proteins that are their own. And this doesn't happen in healthy individuals. And what we found was that for some reason, and we're not quite sure why, individuals with Brugada syndrome created antibodies against 3 cardiac proteins: alpha cardiac actin, keratin, and connexin 43. So in order to have a little bit of a better understanding as to what these antibodies did from an electrophysiological perspective, I created an active immunization model, which on the bottom left, you'll see a little picture of a mouse. Mice were injected with the 3 proteins that we found autoimmunity against, and they were allowed to create antibodies against these proteins. I then conducted ECGs on these mice and compared them to controls. And on the top right, we see 3 graphs. So in the far right, QRS duration, this is a measure of depolarization, so how negative the cells are able to become. And what we note and what we can appreciate is the pink there, being a protein-injected mice, is having a little bit of a longer duration as compared to the other mice. We move to the center graph, QTC duration. This is actually a measure of depolarization and subsequent repolarization. And also, interestingly, in the same mouse, we see that it's lower. And then finally, we look at index of cardioelectrophysiological balance. And this is actually an index that allows us to measure the chance of dangerous arrhythmias happening, like ventricular tachycardia and fibrillation that can lead to death. And interestingly, a low ICEB index is also clinically associated with Brugada syndrome. And once again, we see that it's quite low in the same mouse. We take a look at the bottom black image there, the co-localization of IgG. This is essentially just showing that in the same mouse, that pink mouse, we see that only one antibody actually bound to the surface of heart cells, and that was interestingly connexin 43. Now, connexin 43, that's just an image of that's just an amino acid sequence of the protein. It's a very important protein where 6 of them actually create half of a channel, and you can kind of think of it as one channel on the edge or surface of one cell and another half channel on the other, and connected, it creates a tunnel called a gap junction. And this gap junction is essential for cell-to-cell communication. So for some reason, on our sick mouse, there were antibodies affecting or created against that protein. Next slide, please. And so my question for this puzzle piece of my project is, are anti-connexin antibodies contributing to the conduction changes we see in the mice and also in Brugada syndrome? The objective of the study in this particular part of it is to examine if indeed we can take a look at these antibodies, put them in an environment where we can study their effect on electrical conduction. And one of the ways that we can do this is through an in vitro cell model, or taking a look specifically at iPSC-CMs, which are induced pluripotent stem cells that are coaxed into cardiomyocytes or heart cells. So on the right, we see one of the two techniques, the automated robotic techniques developed by Dr. Houston's lab. And this is actually a dye injection technique where we take a plate of adherent cardiomyocytes and we expose them to something. And in my case, they'll be exposed to connexin 43 antibodies and compared to controls. And we inject cells with a fluorescent dye. And the number of adjacent cells that are actually able to pick up this dye gives us an idea of gap junction intercellular communication. And we can compare this to normal controls. On the left there, what we see is conduction velocity, and this is another measure for and another robotic system that Dr. Houston's lab has developed that we'll also be taking a look at for this assay. And conduction velocity is an important measure of the speed and direction of an action potential. And with a lot of arrhythmias clinically, we see differences from normal in their conduction velocities. So it's going to essentially also be a plate of cells. These cells will be paced and they'll be exposed to connexin 43 antibodies compared to controls, and we'll be able to see using this measure if this conduction velocity is actually affected by the presence. And conduction velocity is cool because not only does it measure gap junctions like the dye, but also gives us an idea as to ion channels, which are very important also in conduction velocity measures. So this is really just one puzzle piece of the large project that I'm doing, but I felt like this was the most applicable to robotics and engineering. But yeah, that's all for me.